This is Chapter 8, Part 3, and we are continuing our discussion of enzymes. And I wanted to introduce the idea of enzymatic pathways, or sometimes called metabolic pathways. And this is referring to the fact that enzymatic reactions rarely exist by themselves in isolation in cells. Usually, enzymes are acting as part of these larger pathways where you may have, 4, 5, 6, 10, 20 different enzymatic reactions that are all linked together in a pathway. So this would be a situation where the product of reaction number one then becomes the substrate for reaction number two by a second enzyme, catalyzed by a second enzyme, and then the product of reaction number two becomes the substrate for enzyme number three, and so on. And there's all kinds of these metabolic pathways that exist in your cells. In this cartoon here, you can see they're outlining, just kind of in a generic sense, uh, pathways that might be involved with lipid metabolism or carbohydrate metabolism, etc., where each one of these colored lines would represent some of these different pathways. And keep in mind, metabolism could be breaking things down or building things up. And there would be examples of each of these in each colored category here. And as you can see, if you were to overlay all these different types of metabolism and all these different enzymatic metabolic pathways on top of each other, it starts to look really messy. And this is really just to illustrate the point that there is a lot of enzymatic activity going on in your cells. So, thousands of reactions happening at any given time. And I also just wanted to point out here, you can see a lot of these different pathways intersect, right? So you can have some intermediate in a carbohydrate metabolic pathway that might feed into lipid metabolism. We could use some component here to help generate lipids or vice versa. So again, it is a pretty complex picture, and this is the kind of thing that biochemists or some cell biologists would be focused on trying to understand, right? How does all this work? How do these things fit together? Um, and it's obviously can get complicated. And we're just going to scratch the surface here. But another thing this illustrates is the fact that if a cell wants to try to maintain a handle on all this complex chemistry that's happening, cells really need to be able to regulate enzymatic activity so they could help, either speed up or slow down specific individual pathways as needed. So that's what I wanted to talk about next, is just kind of the basics of how enzymes could be regulated. And we were talking earlier about partners that might help enhance enzymatic activity, cofactors and prosthetic groups. But here I wanted to really focus on things that might inhibit enzymes. So there are molecules called enzymatic inhibitors that are going to either completely stop or slow down enzymatic activity. And so this inhibition could be irreversible or it might be reversible, just depending on the particular inhibitor and the particular enzyme involved. I'll show you a quick example of each. Here's an example of irreversible inhibition. This particular example is the inhibitor is this molecule called DIPF, which is the active component of nerve gas. And it will irreversibly inhibit this enzyme called acetylcholinesterase. And you may or may not have encountered this one in a past biology class. But the job of this enzyme is to normally bind to its substrate, which is a neurotransmitter that would stimulate muscle contraction. So, a nerve sends a signal to a muscle, hey, you need to contract. This neurotransmitter acetylcholine is released at the nerve ending, binds to the surface of the muscle cell, and that's what's causing the contraction. And then this enzyme, acetylcholinesterase, would come along and chew up any extra acetylcholine that's around, and then that's going to allow the muscle to relax, to stop contracting, because the signal is gone at that point. And so what happens in this situation, if you have this DIPF inhibitor there, it will bind to the active site of that enzyme permanently. So it gets in there and it's going to bind really tightly, meaning that enzyme can no longer bind to its normal substrate. So it basically completely inactivates that enzyme irreversibly. And, in terms of the physiology now, the reason nerve gas is so scary is because it's basically going to cause your muscles to stay contracted once they do contract. They can't relax. So you could think of this as kind of you'd be in a situation with basically full body permanent muscle cramps, right? To the point where, key muscles, diaphragm, heart muscle would freeze up. So that's a scary example, but of course there are irreversible inhibitors that are not, toxins that your cells might need to use in particular instances. Then there are also reversible inhibitors, so this might be something that could bind to the enzyme and inhibit it temporarily, but eventually the inhibitor is going to fall off and the enzyme could resume its normal activity. So irreversible versus reversible inhibition. And I also wanted to point out this idea of inhibitors could be competitive or non-competitive. And the examples we've seen so far have been competitive inhibition. And what that term refers to, it means the inhibitor is going to compete for binding at the active site, compete with the substrate for binding to the active site. So just like we saw with the DIPF nerve gas example, right, that inhibitor was actually sitting in the active site and outcompeting the substrate. So if the substrate can't bind to the enzyme can't catalyze whatever reaction it's supposed to carry out. So that's competitive inhibition. That kind of makes sense. Non-competitive inhibition would be a situation like this, illustrated here, where the inhibitory molecule binds to the enzyme, but it does not bind to the active site. And you might think, well, okay, if it's not binding to the active site, then no big deal, right? How can it prevent the enzyme from working? And the way it would do that is as you see here, it will cause a shape change in the enzyme when it binds. So even though this little green molecule, the inhibitor here, has not touched the active site, when it bound to the enzyme, it caused the active site of the enzyme to change its shape, so substrate can't bind properly, and the enzyme obviously can't do its job properly. So inhibition could be competitive, could be non-competitive, could be reversible or irreversible, just depending on the particular situation that we're dealing with. And one of the main classes of inhibitors I wanted to talk about are allosteric inhibitors. And the reason I wanted to emphasize these is because we're going to see some key examples of allosteric inhibitors working in some of the upcoming chapters. So when we talk about cellular respiration in the next chapter, there are some of the key enzymes in glycolysis and in the citric acid cycle are regulated allosterically. And so what does this mean? Well, this term allostery literally translates as different shapes. So these are enzymes that can exist in usually two different shapes: an active shape, where the enzyme can bind to a substrate, catalyze the reaction, and then an inactive shape where it can't. And allosteric inhibition is always non-competitive. So allosteric inhibitors are not going to bind to the active site. As we were just talking about, they're going to bind somewhere else on the enzyme and change its shape. Another key characteristic of allosterically regulated enzymes is they usually have quaternary structure. And if you remember back to our discussion of macromolecules and proteins. We went through those 4 different levels of protein structure. And quaternary structure was something that only some proteins have, and this referred specifically to proteins that are made up of multiple polypeptide chains. So that's usually the case with these particular enzymes. And I think I'll just show you the image here. So here's an example of an allosterically regulated enzyme, just kind of a generic cartoon. And you can see the active site is up here in what's called the catalytic subunit, which is kind of the light purple subunit portion of the enzyme here. And we can see up top here there's these 2 different shapes, 2 different forms of the enzyme. Again, that's that allostery. So we have the active form where we do have this active site where substrate combined, and then on the left we have the inactive form where the active site is basically gone, so obviously it can no longer bind to substrate. And what you see down here on the bottom is the role that these regulatory molecules are playing. So on the left, you have an allosteric inhibitor that's binding non-competitively to the enzyme and basically kind of, at least temporarily, locking it into that inactive shape, right? So no active site. Whereas on the right, you have an allosteric activator molecule present, and it's helping push the enzyme into that active shape. So this provides a nice opportunity for the cell to either turn on or off enzymatic activity, right? If the enzyme's cranking away, catalyzing this reaction, and the cell wants it to stop, it can simply start producing this inhibitor, or vice versa. If it's stopped and it wants it to kick on, we produce the activator. All right, two final points here. So all these examples of regulation that we've talked about so far have been just kind of at the individual enzyme level. But I also wanted to point out that you can have regulation at the kind of a larger pathway level. And so what we're seeing here is pretty common, and this would be called feedback inhibition, or sometimes called like a negative feedback loop. And what we're looking at down here in the bottom is one of these metabolic pathways that I was describing earlier, where each green line is a different chemical reaction that's catalyzed by a different enzyme. So the first reaction in this particular pathway utilizes the amino acid threonine as the substrate. An enzyme that acts here at the first green line would convert threonine into alpha ketobutyrate. And then a second enzyme would convert that into something else. And a third enzyme would convert that into something else, and so on, until we get to the last reaction and we end up with isoleucine, which is a different amino acid, as our product. So basically the role of this pathway is to convert one amino acid, threonine, into another. And the feedback comes in here because the end product, isoleucine, is actually capable of acting as a non-competitive inhibitor of the first enzyme up here at the beginning of the pathway. And so this is kind of nice for the cell, because you can imagine what's going to happen. Let's say the cell has tons of threonine present, right? And all these different enzymes are available in the cell. That threonine is going to start to be converted into isoleucine by this group of enzymes. And as the isoleucine levels build up, some of that isoleucine that's floating around is going to bump into this enzyme that's catalyzing the first reaction and start to shut it down. And so as we shut that enzyme down, basically this whole pathway begins to come to a grinding halt, right? And so eventually, if there's enough isoleucine, we basically shut down the whole pathway. Okay? And so then what's going to happen is, well, if the pathway's shut down, then we're not making any more isoleucine, so isoleucine levels will start to drop off, right? Isoleucine will get broken down or utilized by the cells. It'll start to disappear. And as it goes away, then this enzyme is no longer inhibited, and this pathway could kick back on. So it's kind of like an automatic on-off switch, essentially. Um, there are also examples of like positive feedback loops where the end product of a pathway could actually stimulate, enhance the activity of some of the enzymes in that pathway. But they tend to be, those positive feedback loops tend to be a little bit rare in terms of biological systems, whereas this type we're looking at here, negative feedback, is very common. All right, last point I wanted to make is just to remind you that because enzymes are proteins and proteins are sensitive to environmental conditions, you might remember we talked about this in an earlier chapter, right? Proteins could be denatured by high temperature pH, meaning the three-dimensional structure of the protein will break down, right? It basically starts to unfold. And if that happens to a protein that's an enzyme, because enzymes are really dependent on having the correct shape for their biological activity, if we start to unfold the enzyme, it's not going to work anymore. And so, in terms of practical biology and biochemistry, what that means is every enzyme is going to have an optimal pH where it works best. And that may be different for different enzymes. Here you see 3 different enzymes that have 3 different optimal pHs. And every enzyme is going to have an optimal temperature where it works best. And that's just because, for example, for chymotrypsin at this particular pH, about pH 7.5, that is the pH that's going to allow that protein, that enzyme, to fold correctly and have the correct shape. So it'll be most efficient at catalyzing its particular reaction. Same idea with temperature. And we'll explore that idea a little bit more in lab as well. So anyway, that is the end of Chapter 8. We talked about some basic energy concepts, talked about delta Gs and what that means in terms of reactions releasing or consuming energy. We talked a little bit about ATP. We talked a little bit about some of the basics of enzymes and enzymatic reactions and how those could be regulated.